Hi, girls. In the earlier session, we saw about the various parts of bacteria and their function. And this session also will be the continuation of the various parts and the function of bacteria. In this session, let us discuss about the locomotion of bacteria, the mode of respiration, nutrition, and reproduction in bacteria. Nutrition of bacteria. Bacteria need enough suitable nutrients for its growth and reproduction, enough moisture and temperature, suitable pH, and enough nutrients such as carbon, nitrogen, and trace elements are necessary for the growth of bacteria. Bacteria are diverse in their specific nutrient requirements. On the basis of nutrition, bacteria are divided basically into two groups, namely autotrophs and heterotrophs. The bacteria which utilize carbon dioxide as a source of carbon are known as heterotrophs. Autotrophic bacteria. The autotrophic bacteria use sunlight as the energy source and they are called as photoautotrophs or photosynthetic bacteria. Example, green sulfur bacteria. They are capable of synthesizing their own food. The autotrophic bacteria, which derive energy by oxidizing inorganic compounds, are known as chemoautotrophs. Example, nitrifying bacteria, sulfur bacteria, and iron bacteria. The nitrifying bacteria derive energy from nitrogen, sulfur bacteria from sulfur, and iron bacteria from iron. Here we can find the images of the autotrophic bacteria. The green sulfur bacteria, which is autotrophic, you can see the green color, and therefore we know that there is a green colored pigment capable of synthesizing their own food, and the nitrifying bacteria. Autotrophic bacteria. The nitrifying bacteria oxidize ammonium salts into nitrates to draw energy to make food from carbon dioxide. Likewise, the sulfur bacteria oxidize hydrogen sulfide to get energy from chemosynthesis. The iron bacteria oxidize ferrous iron compounds into ferric hydroxides to get energy. The carbon bacteria in the damp coal oxidize carbon monoxide into carbon dioxide to derive energy. Heterotrophic bacteria make use of organic compounds such as simple sugars and amino acids as the source of carbon. The bacteria, which lives in the cells of other organisms by feeding on the cellular compounds, are called parasitic bacteria. Those living on dead organic matter to draw energy and carbon are called saprophytic bacteria. The bacteria which live in association with other organisms are known as symbiotic bacteria. Example, rhizobium present in the root nodules of leguminous plants. Some heterotrophic bacteria use sunlight as the energy source and organic compounds as the source of carbon. Such bacteria are known as photoheterotrophs. Example, non-sulfur purple bacteria. Some heterotrophic bacteria use organic compounds such as carbon as well as energy. Such bacteria are known as chemotrophs. Bacterial respiration. The bacteria may be aerobic, anaerobic, or facultative anaerobic. Aerobic bacteria use oxygen for respiration. Anaerobic bacteria use carbon dioxide. Facultative anaerobic bacteria use oxygen when oxygen is available and use carbon dioxide when oxygen is not available. Aerobic bacteria. Aerobic bacteria require oxygen for growth and can be incubated in an air atmosphere where the oxygen is about 21%. In this image, we find the growth of aerobic bacteria in a culture. Anaerobic bacteria. Anaerobic bacteria do not use oxygen to obtain energy. Moreover, oxygen is toxic for them, which means the bacteria dies in the presence of oxygen and they cannot grow when they are incubated in an air atmosphere. Some anaerobic bacteria can tolerate low levels of oxygen They are called as tolerant anaerobes, but others, which are the strict anaerobes, cannot tolerate even low levels and may die upon brief exposure to air. We can find here the anaerobic bacteria. Facultative anaerobic bacteria do not require oxygen for growth, although they may use it for production if it is available. They are not inhibited by oxygen and usually grow well. Microaerophilic bacteria, they require low levels of oxygen for growth but cannot tolerate the level of oxygen present in the atmosphere. Here we can find the different types of bacteria, the aerobes, anaerobes, facultative anaerobes, and tolerant anaerobes and microaerophiles. Temperature tolerance of bacteria. Based on temperature tolerance, the bacteria are classified into different groups, namely mesophilic bacteria, thermophilic bacteria, and psychrophilic bacteria. The mesophilic bacteria grow well in temperature between 25 degrees centigrade and 40 degrees centigrade. Thermophilic bacteria grow well above 40 degrees centigrade, while the psychrophilic bacteria grow well in temperatures less than 25 degrees centigrade. So they can also grow inside our refrigerator. Bacterial reproduction. In bacteria, growth and reproduction are simultaneous phenomenon. Bacteria reproduce mainly by asexual methods like binary fission, budding, fragmentation, endospore formation, and the formation of conidiospores. Binary fission. Most of the bacteria reproduce by transverse binary fission. A single bacterial cell divides into two cells by the formation of a transverse septum. Each time a cell divides, they form a new generation of two cells. Each cell of the new generation is capable of further division by fission. In this image, we can find the process of binary fission. The presence of nucleoid, the cell replicates its DNA. So we have two masses. The cytoplasmic membrane elongates and they separate the DNA molecules. Cross wall form between the two DNA molecules. There is invagination and cross wall forms completely and they too separate and form the daughter cells. Here you can find the beautiful process of binary fission. A single bacteria dividing into two daughter cells. Budding. The genera Pasturia, Blastobacter, Ciliberia, and Pantomyces are generally known for budding bacteria. Budding involves development of a small protuberance bud at one end of the cell. This enlarges and eventually develops into a new cell. The new cell cuts off from the parent and grow as a new individual. This is the budding process. So in the first, we find that a small bud is there and then there is the transport of the nuclear material, formation of the transverse wall, and sometimes budding process produces chain of cells. Here you can find the budding process, the process of budding in the bacteria. So a single cell has divided into two cells. There is a cleavage formed between the two cells and they divide into two daughter cells. They have different set of the nucleoid and cytoplasm. Fragmentation. Bacterial genera such as Saprospira, Dichothrix are filamentous in nature. They usually reproduce by fragmentation of filament. As a result, short filaments are formed and they grow into new filaments. You can find the fragmentation process taking place rapidly and beautifully. Endospore formation. Endospores are bodies produced within the cells of the bacteria. During unfavorable conditions, the nucleus duplicates and the protoplasm secretes a thick wall around the nucleoid. This thick walled structure is called as the endospore. During favorable condition, it grows into a new bacterium. They are more resistant to unfavorable environmental conditions such as heat, cold, desiccation, osmosis, and chemicals than the vegetative cells. Example, Bacillus and Clostridium. Spores may be spherical, ellipsoidal, or cylindrical in shape. The position of the spore in the cell may be central in the center of the cell, eccentric in one pole of the cell, subterminal or terminal. Each species has its own characteristic size, shape, and position of the spore. This is how the endospore is formed in the bacterial cell. You can now watch the endospore formation in the bacteria. You can see the endospore that is formed within the body of the bacteria. A cortex is formed. There is a protein coat surrounding the endospore, the exosporium, and this is released. The mother cell degenerates. So this endospore is capable of growing into a new cell when there is a favorable condition. This again will illustrate the formation of the endospore formation. Inner spore membrane, outer spore membrane, formation of the cortex. Exosporium has now formed and the bacterial cell disintegrates and liberates the endospore. The endospore is dormant and it will grow whenever the environment is conducive. Conidiospores. Streptomyces-like organisms produce many spores by developing cross walls or septa at the hyphae tips. Each spore gives rise to a new bacterium. You can find here the conidiospores. And this image also shows the formation of conidiospores. In this session, we saw the locomotion of the bacteria, mode of nutrition, the temperature at which the bacteria can thrive, about the nutrition of bacteria, and also on the different methods of asexual reproduction present in bacteria. Thank you.